Hello, everybody. My name is Russey. I'm from the Institute of Forestry Science in Madrid. And yeah, I'm excited to present to you my research. I'm in the first year of my doctorate, and this is the first time I'm presenting my work. I do have to say it's a little bit data heavy, so I hope you will not get too bored. I tried to make it as understandable as possible. Yeah, so I already had some of my colleagues here already introduced the topic a bit on the remote sensing. So yeah, the technology for forest inventory and phenotyping has changed a lot over the last decades, especially. And yeah, so we will be using, in my work, we used a lot of the newer kinds of technologies in LiDAR and with multispectral imaging. And so we wanted to find the genetic parameters in provenance clonal test for the maritime pine and find the relationships between the genetic traits. And then we want to try to apply these to low input breeding programs to breed like more resilient pine trees and pine forests. So the genetic material we selected from 34 provenances across the natural distribution of the maritime pine. From there we took seeds and we grew these into seedlings and then took cuttings from these so that we have basically clonal organisms from every one of these sites, and so there is no genetic difference between any of the individuals from each site. And then we planted all of them in a common garden trial in Asturias, in the north of Spain. It's the black square in the top there, named Armayan. It's a very beautiful place, so I recommend you to visit the north of Spain. And yeah, after some time when they were at the right height, around 17 years old, the trees, we went with our team to collect the data. We used terrestrial LiDAR and UAV mounted LiDAR and also multispectral imaging to get most of the data. So then we did a lot of data analysis. That's the part where I was more involved. So we did normal distribution tests to check the data. Then a random forest model to determine some of the values we couldn't get from from the direct data collection. So we could extrapolate from the data we got using this forest model. We could extrapolate the volume and the biomass. And then we tried to use best linear unbiased predictors to isolate the genetic component, so we didn't have to think so much about the environmental component at first. And then using these blobs, as we call them, we did a principal component analysis to find correlations between the genetic traits. And then finally we did a heritability test using pedigree charts to see how heritable the traits are. Yeah, so these are all the variables that we got at first. So we got from the terrestrial LiDAR the normal ones, like the diameter at breast height, the total height and inclination of the trees. And with the aerial LiDAR, we found more the understory and the upper canopy density, so the forest structure, and then the diameter of the crown as well. And then with multispectral imaging, we found a lot of different variables that were related to the health of the plants and how much stress they were under and these kinds of things. Yeah. So here's some of the results. This is on the population level. You can see there's not so many dots. On each of the dots is one of the populations you could see on the map earlier. Yeah, so you can see most of it is rather intuitive. So we found the tall The taller trees had more upper canopy density, which makes a lot of sense. Then you can see there is a little groupation of data there of traits there that are related to the plant health. So means more or less that the healthier plants are growing taller, which makes also sense. And then on the bottom right there, so those traits are from the multispectral imaging and show more or less the stress levels the trees are under, and these are then the populations that were under more stress were tended to be less tall and with less upper story growth. So here we can see on the clonal level, sorry, the image is a bit low resolution, that the data is a bit more spread out, so the traits are a bit less directly correlated. And yeah, we can see that it's more or less very similar. There's relatively tall growth is related to, again, upper canopy density and more inclination of the trees. And then the understory, lower trees and the more understory growth has to do with usually have more stress and more chlorophyll content, which makes sense because they would be competing for the light underneath the taller trees. Then in more of the data analysis, the normality, we found that, so not all of the variables were normally distributed, and to determine the cause of that, we still need to do more analysis because we are still in the data analysis section. We don't have like conclusive results yet. It's more, preliminary. And then with the random forest model explained most of the variance for the total volume. So later on we will be using the total volume, yes, but the biomass, we still have to improve the model to get better results. Then in the heritability tests, you can see the most important part is the significance, because this means that if it's significant, then this trait is highly heritable. And so, what we found that almost all of the traits were very highly heritable. So, this is a good thing that we can use for the breeding programs later on. And we still have a lot of work to do. So we still have to do a lot of data collection to get more robust data. And we also have to add the environmental part of it and add the provenances and the locations that they were from, so that you saw on the map earlier, to really make sense of which populations we will select to further breed in breeding programs. And thank you very much.